Okay, so I thought I might try creating a few short videos, which basically kind of summarize some of the Substack posts. Perhaps people prefer video content more, I don't know. So I start with Nautilus. Um, so I wrote this in 2021, and I haven't really caught up with Nautilus again since then, but I'm just going to summarise what I wrote about then. So the background of the company is kind of interesting in that the CEO comes from Iceland, which is a more traditional technology company. It's a data storage company. So Iceland built these large storage systems which let you store kind of petabytes of data. And they were very popular in the genomics community, and I know a number of startups who purchased Iceland solutions because they were very easy to use and they would scale out, and you didn't need to spend a huge amount of time supporting those storage systems. And they also did a deal with Illumina, so I kind of guess this is how the ex-Iceland guy got involved or got to know about Nautilus and moved into biotech, is by seeing that there were these large storage applications. Nautilus had three main areas of innovation which they discussed initially, and I kind of found in their patents. The first is they have this method of arraying single proteins on a surface. Then they talk about a method for identifying and fingerprinting those proteins when they're on the surface. And they have, or they talk about approaches to developing a library of affinity reagents which they use in the fingerprinting process. So the first thing is, like, they've got this way of ordering the proteins on the surface, and this almost seems like the core technology. This seemed to be at the time the thing that was most thought out. On the left side of the screen, you'd see what happened if you just threw molecules down on the surface. They'd all just go down randomly, and you'd get some sitting on top of others, so you couldn't really clearly distinguish them, and you'd get very few, or you'd get some percentage of single proteins that you could clearly distinguish, and you wouldn't really be making maximal utility of use of the kind of surface. And on the right, you've got an ordered array where everything is nicely laid out, and it's much nicer to see the individual molecules. Technologies like Illumina's DNA sequencing platform went through this evolution from an unordered kind of system through to an ordered system. So generally the platform will start with an unordered array, which is kind of not making best use of the surface, but you build a platform that works. And then after you've got the system basically working, you'll move towards an ordered array where everything is optimally laid out. You've proved out the technology, and then you now want to push throughput and density of the platform. Nautilus have started with an ordered array, and one of the things I couldn't quite figure out is, well, why would you do that initially? It doesn't seem like the most important problem to work on, but it seems like the most developed thing, which is a bit weird. So this is an image from one of our patents showing how they do it. Basically, they create DNA nanoballs. So these are similar to the nanoballs that are used in MGI sequencing or rolling circle amplification. They do some level of size selection, I believe, on those nanoballs to get that, so they only get them the right size. And they've got kind of landing pads that these balls can attach to for charge-based attraction on the flow cell, and these nanoballs have a site to which a single protein can be attached, which is cool. So it's a single molecule process, but they're using these amplified balls to do the attachment. So then they have to do something with the proteins, and this felt like in the patents at the time was the least well thought out part of the process. Essentially, it just says you flow things in antibodies, aptamers, whatever, and they will attach to your proteins, match some kind of motif or structure on your protein, and they will be fluorescently labelled and they'll light up, and you'll do that over a number of cycles. And with all that data plus AI, you'll get a unique signature out for each of your proteins, which you can use to identify which proteins you see in the relative abundance of various proteins in your sample. So alongside that, they've got this IP around developing libraries of affinity reagents, in particular aptamers, and it's basically felt like a pretty traditional aptamer development process, but maybe you can use your single molecule arrays to help out with this, and they show some examples of that in the patent. And then they also have this idea of developing affinity reagents that specifically target like various different femurs or tumors in your protein, so you're going to get unique labels for each tumor or fiber or whatever. You can kind of combine them. Again, not massively built out. So I think that was it really for Nautilus. There's a bit more information in the Substack post, which if you want to hear a bit more about the approach and my thoughts on it, you can go and refer there. They reached out a while ago to have a chat and say maybe answer some of my questions and give an update, and I didn't do that, but I should probably go patent hunting at least again at some point and see what they're up to. But if you have any thoughts on Nautilus, I'd love to hear them, or please get in touch either by email or on the Discord or whatever. Yeah, that's it for now.